As I introduce our first guest this afternoon, one difference we have today from previous years perhaps is the high level of enthusiasm and participation and even attendance from some of the other individuals in the field who are not even presenting today. That includes Gino Cortopassi here from UC Davis, who's involved in drug discovery and FRDA. Dalia Megiddo from BioBlast, who's interested in protein development replacement therapies, as well as a variety of individuals around the room from ViroPharma who are working on developing OX1, who I know I was very impressed with their participation at yesterday's ride and the other things that they're doing for us at CHOP as well. Which allows me to now direct my introduction to my friend, Mr. Jim Rusche from Repligen Pharmaceutical. As you know, or almost everyone in this room knows, Repligen has been intimately involved in the development of histone deacetylase inhibition, HDAC inhibition, since it was initially discovered by Joel Gottesfeld and then licensed to them, about 5.5 short years ago is my recollection. Jim's here to tell you about some of the successes, some of the difficulties, and where we go from here. Jim.
Hello. So let's see. So as Dave said, I'm going to try and tell you about the work that we've done to identify a development candidate for Friedreich's ataxia. We did start on this work about 5 years ago and have had significant collaborations from the start with Joel Gottesfeld and with FRDA. And that's helped us advance at a pace that we could not have accomplished without that help. So let me start by going through some of the background that we were observing when we got started in this program and how we tried to develop a rationale for the development of these experimental compounds. So some aspects that probably most of you know, but let me go through them a little bit just to describe the molecular basis for the disease, which gives us some insight as to what we might do to treat the disease. So the cartoon on the left shows, I see you can't. On the left, you see a cartoon of a gene, and it indicates that there's a variety of different triplet repeat diseases. Those triplet repeats cause a disruption in the normal organization of that gene. It can occur at the beginning of the gene, and as a consequence, the gene is not transcribed properly. It can occur at the end. It can occur within the coding sequence of the gene, and that's the case in Huntington's disease. But in Friedreich's ataxia, this triplet expansion actually occurs in a non-coding region inside the gene called an intron. This is the part that's normally spliced out when you go from DNA to make RNA, and then the RNA codes for the protein. The mature RNA doesn't even have this region. The problem is with the triplet repeat expansion in the frataxin gene, the RNA starts but never is able to complete its way through the gene and make a complete RNA because this triplet repeat expansion causes the DNA to take on an unusual conformation. That's the theory. And the consequence of this unusual conformation in the intron 1 triplet expansion is that the normal amount of frataxin that's made off of the RNA is decreased significantly because there's a lot less full-length RNAs that contain all the exons. And so the problem is one needs to not change the ability to start making the RNA, one needs to change the ability to make full-length RNA. And to understand how we might do that really takes some understanding of what's going on in this region of the gene and why it is that the transcription of the DNA into RNA is not occurring the way it should. One of the things that's important for us when we think about what is the goal of our therapeutics development is to realize that this is an autosomal recessive disease, and if one examines heterozygotes, somebody that only has one copy of this triplet repeat expansion, they actually do make about half as much frataxin, RNA, and protein. but there's no apparent disease. So that tells us that that's the goal that we want to try and achieve. We want to try and achieve the expression levels for which natural history tells us there isn't going to be a physiologic consequence to the neurons for the reduction in the frataxin protein. So one of the important observations that Joel Gottesfeld's lab made 5-6 years ago was the understanding that what was the organization of the frataxin gene when it had this triplet repeat expansion. And what he was able to discern is that it had the structure and it had the characteristics of what's called heterochromatin. Heterochromatin is that region of our genome, that region of the chromosome that's not really expressed. So it had taken on some conformation and it had all the hallmarks of unexpressed region of DNA. And he characterized what that looked like. So the cartoon here at the top shows a structure of a nucleosome. So this is made up of histones 1, 2, 3, and 4. And these histones form a ball and make like a bead where the DNA wraps around it. And then that just continues for thousands and thousands of base pairs and nucleosomes, like beads on a string. And the organization of this DNA around the histone is controlled by how that histone protein is actually modified. And it's modified by a number of post-translational modifications. So after the histone polypeptide is made and assembled into this 4-protein ball, its tails get modified. They might be methylated. They might be acetylated. They might be phosphorylated. And Joel and his lab looked at this pattern of organization because it's this nucleosome that controls to a large extent how that region of the DNA will be expressed. And what they found was that the pattern of changes, the code that tells the genome that this is a region that should be expressed and should be transcribed from DNA into RNA, that code was changed. And these acetylation patterns that normally exist throughout the histones had changed. And most specifically, and this has taken a few years of work, at this particular position on H3 at a lysine, so H3K9, there's more methylation and less acetylation. So this particular notation on the histone in the position of the frataxin gene is thought to be a consequence of some of the structure that that region takes, as I was saying before. And exactly how that structure makes the code change on the histone is something that's being actively studied, but nobody knows exactly. But what this tells us as we go to develop therapies is, gee, if we could return this code to the code it was before, we may be able to get this gene to be transcribed as it was before. And that's the basis for trying to modify, using histone deacetylase inhibitors, the code or the notation that's on the histone. So histone deacetylases take off this acetyl group. And when they do so, then it allows more methylation to take place. So what we wanted to do was to decrease the ability of these histone deacetylases to make that change. And if we inhibit those enzymes, the acetylation stays put. So there's more acetylation at the H3K9 position. So that's the basis for which we're developing small molecules to inhibit these enzymes. Now, the histone deacetylases is a group of enzymes that are highly related, but each one is different, and over time, evolution has made a group of about 18 enzymes. The groups that are of most interest for the control of chromatin and the structure of these histones appears to be the class 1 group. HDACs 1, 2, and 3 are nuclear in their location within a cell, and they seem to be more important for the regulation of gene expression. And prominent within neural cells is HDAC3. The compounds that we work on are ones that are specific for HDAC 1, 2, and 3. And so we have focused on those, and that's supported by the fact that some of the first generation compounds that were looked at were compounds that were active against HDAC 1, 2, and 3, and most active against HDAC 3 to inhibit that enzyme. And so that was the basis by which we focused on this class of compounds. Now, structurally, the chemical structures that have been identified as small molecules inhibitors of these enzymes all have some things that are quite similar. They all like to bind in the active site of the enzyme, and in that active site is a metal, a zinc. And all of these compounds have a similarity in that they all like to go into that active site and bind zinc. And when they do so, they disrupt the ability of the enzyme to be a deacetylase. So on an atomic level, they're stopping the transition for this enzyme to be able to catalyze the deacetylation of the histone. So very simple compounds like valproic acid do so with a alkyl group and an acid group. Hydroxymates do so because the hydroxamic acid can chelate. In the benzamides, the compound class that we've worked on, this orthoamine amide group right here is also able to interact specifically with zinc. And then cyclic peptides, which started out as natural substances and since then have been synthetically made, also interact with a zinc, oftentimes through a sulfur group. So the compound group that we're focusing on, the benzamides, has historically been used as anti-cancer compounds. So there's 2 of those that are currently in advanced clinical studies. But none of them have been developed, nor are they really suitable for use in diseases like Friedreich's ataxia. And we've worked using medicinal chemistry to synthesize many derivatives of these class of compounds to find ones that we think have the effectiveness and the lack of toxicity that you'd want to use in this kind of a patient population. And a compound we've identified and advanced in development is called 2833. So just a few things that we've learned in our labs and in collaboration with a number of others, including Joe Gaddisfeld's lab and Massimo Pandolfo's lab, and through a great deal of help from the FARA organization, we've been able to identify a few important features of compounds that work to increase frataxin. At first, it was thought that maybe HDAC3 was the important enzyme and that the best thing would be a compound that was specific for that enzyme. That's the magic bullet. But it turns out that that's not the case. It turns out that if you have a compound that's really specific for HDAC3 or HDAC1, neither of those compounds work. But compounds that are active against both of those enzymes work. So that was an important fork in the road in finding good compounds for drug development for this disease. We look at these compounds and their potential impact on other neurodegenerative diseases as well. And for example, in Huntington's disease, an HDAC3 specific compound is active both in cell culture and in animal models. So it's really important to understand the molecular basis of the disease and the focus of the compounds that you're trying to develop. So an important tool for us in understanding compounds that work and how they work has been to be able to use primary cells from patients. Fibroblast cell lines that have been made and lymphocyte cell lines that have been made do not seem to work in culture as reproducibly as primary cells. So an important tool for us has been to be able to work with patient advocacy groups and the patient community to get primary cells. So blood donations are provided to us. We're able to isolate peripheral blood mononuclear cells, and those in culture can then be treated with different compounds, and we can determine different important features of frataxin expression and increases in expression. So this has been a very important partnership for us in being able to tell the difference between, in hundreds of compounds, we make good ones from bad ones and trying to develop, obviously, the good ones. These compounds have also been used in animal models. While the animal models for Friedreich's ataxia are not the most sophisticated and accurate in being able to reproduce the human disease, there are some mouse models that have decreased expression of the frataxin protein. We can treat them with the compounds and see whether we increase the expression of that protein. And along the way, something else we learned is that while histone deacetylase inhibitors can change the expression of a number of different genes, it's not just the frataxin mutant gene. One thing that we learned was that the wild-type frataxin gene is not really affected by these HDAC inhibitors, but the mutant gene is. And that, I think, is consistent with the fact that the frataxin gene is not somehow deficient in initiation, which is often changed by HDAC inhibitors, but in elongation, which is something that is a different property of this gene and is affected by these HDAC inhibitors. So this slide just shows an example of a dose response. So on the left panel, you see increasing amounts of 2833 and the increase in the frataxin RNA in cell culture as a result. These are using patient lymphocytes. Whereas if you take control cells, you see a small, very small increase in the amount of RNA. And in the right panel, you just see that similar to the increase in RNA, there's an increase in the amount of protein. So we've had the fortune over the last couple of years to be able to do this experiment many times. And this has helped us answer a number of different questions. One of the questions that you want to answer is, okay, so do 10% of the people respond to this drug? Do 100% respond to the drug? And a way to do that was to look at enough different patient samples to see if we could understand what the response rate was. So at the bottom, you can see with an N of 47 that an expression level that we can measure. So anything below 1.3-fold increase, and our techniques are not really able to discern the difference between increase and no increase. So that's kind of the floor of what we can measure. And that about 83% of those 47 patient samples showed an increase. And if you look at a 2-fold increase, that would, on a theoretic level, bring them to the same level of a heterozygote that has no disease expression, that there's about a 50% response rate. So this looks and mathematically shows that virtually all samples will show a response, but that response can be from something that's quite small and unlikely to be a therapeutic change to something that's significant and would likely be a therapeutic change. Now, this leaves unanswered many questions. If you take a single patient and they were to be treated with drug once a month for a year, how many of those 12 times would they actually show this response? Every time? Half the time? So it's just a different way of asking this same question of what is the response rate to the drug? And we don't know the answers to those questions. It's only through further work and through clinical studies that we'll be able to address those kinds of more sophisticated questions. But it's optimistic that such a high percentage respond to the drug. This just shows that another tool that we have, again, developed in Gottesfeld lab, is to use not lymphocytes, But since this is a neural disease and a neuromuscular disease, we wanted to look at neurons. And the Gottesfeld lab has been able to make from patient cells pluripotent stem cells, and from those stem cells to then differentiate them into neurons. And when you do that and you repeat these cell culture experiments with neurons, you see the same thing that we saw in lymphocytes. So you see a dose response to increase the amount of frataxin. If you use a compound that's specific just for HDAC 1 and 2, you don't see much of anything. And again, the protein levels increase against controls, as you would hope. So it's good to know that this is not some artifact of looking at blood lymphocytes, since that wouldn't be very therapeutic on its own, but that we actually can see the result in neurons. Another aspect of using these kinds of HDAC inhibitors is to think about how one might provide treatment. And this is an experiment that was done in mice, but it points out an important feature. The blue curve is measuring the amount of drug in the brain. And you see it goes up and it comes down rather quickly. But the point is that once you've put the compound in, and it starts a cascade, and that cascade is the enzyme gets inhibited, then you get an increase in the acetylation, and then you get an increase in the expression. So we don't know exactly what that timing will be in man, but it's an important feature that we would not dose this drug for a constant exposure, but we would dose the drug to try and induce a change that would then persist for some time after the drug. So intermittent treatment is the kind of designs that we're trying to evaluate now when we think about safety and efficacy, is to think about this property of intermittent dosing. One of the things that we did early on in cell culture with Giovanni Coppola at UCLA, Joel's lab, and others, was to look at what the signature across the entire genome was for the expression of genes. So using peripheral blood from 10 patients, 10 carriers, heterozygotes, and 11 controls, isolated that, incubated them in culture with drug, and then looked at the expression of all the genes, not just frataxin. And what we saw in that is that there was a subset of genes that were different in patients compared to their related carriers or family member. And those 77 genes, in essence, represented by this criteria a disease signature. So these were common genes that were changed in frataxin patients. What we then did was to look at what did the drug's effect have on these genes. These are genes that are probably changed in their expression because a cell has not enough frataxin protein. So these are gene changes that are a consequence of not having enough frataxin. And so what we saw was when you looked at these 77 genes, so each row represents one of those 77 genes, and this represents the 10, each column represents the 10 patients, or patient carrier pairs, I should say. So you can see that there's genes that are increased in expression and those that are decreased in expression. And upon drug treatment, you really get a reversal of that. What that represents is that the gene expression change, which is, if you will, the signature of the transcription in the disease, is reversed more towards a normal transcription pattern. And that's what you would hope to see if the change that you were inducing in frataxin expression had a therapeutic benefit to the cell. So that's something that we will endeavor to follow in the future as well. So I am doing all right? Compounds, any compound, and these compounds are no different, are not without potential toxicities. And the compound class of HDAC inhibitors has some toxicities that have been seen across a number of these different compounds. Certainly in cancer, where they try and treat with a drug at its maximum tolerated dose, its MTD, you see a lot of toxicities. We're trying to do something different. We're trying to dose below any observable effect level of toxicity. So there's a real difference in how the compounds are used. But when we do toxicology studies in animals to help us understand and prepare for exposures in man, we push the drug levels as high as we can, and then we can see these toxicities as well. And some of those toxicities include lymphoid atrophy and inhibition of spermatogenesis. Those are both reversible. That is, when you stop dosing, those toxicities reverse and are gone. But that gives us an idea of what kind of levels are needed to get a toxic effect. And I don't need to tell you that this is a very debilitating disease and can be lethal. And in the absence of any treatments, we need to balance how to go about developing such a compound with doing so in a safe and responsible manner. And hopefully through our efforts, yours, and the FDA and other regulatory agencies, we can find that pragmatic path to understanding how to develop these new treatments where there are unanswered questions for both safety and efficacy. So what have we done in the way of development? We have taken this 2833 forward into clinical development and tried to ask a number of questions. Because we have the ability to measure in peripheral blood lymphocytes what changes there might be in frataxin gene expression, that's one of the goals that we had in early clinical studies. Obviously, the primary goal of early clinical studies is safety. The second goal is pharmacokinetics. Then you can explain how much drug exposure there was and try and relate that to other properties. But in our case, being able to measure frataxin gave us a third goal, and that's a biomarker. We wanted to see whether or not at safe and tolerated doses, oral doses of HDAC inhibitor, we could get an increase in frataxin expression. That's our proof of principle. That's what we thought we could do. We've done in animals, but that became the goal to do in man. One of the important tools that we needed to do that was to be able to accurately measure frataxin protein in blood and to understand over the course of time, did everybody's frataxin protein change day to day, or was it relatively stable? If it changed day to day on its own, then any change our drug might induce would be difficult to see. So in conjunction with a family advocacy group in Italy, GOFAR, we collected blood samples from 5 patients and 5 carriers once a week for 15 weeks. And then we measured the blood levels and the RNA levels. We measured the RNA and the protein levels in blood, and you can see the result of that test here. And they're relatively stable. And so it's going to be relatively easy for us to discern a significant change in the expression of the frataxin gene. And it's in this backdrop that we'll be trying to examine the consequence of drug treatment in patients. So our development efforts over the last couple of years are summarized on this slide. In the United States, we filed an IND in 2010, and the FDA put us on clinical hold. They felt that the information that we had provided to them did not adequately characterize the potential safety issues with the drug. They wanted to make sure that any of the toxicities that we saw were reversible, and they wanted us to make sure that we didn't perform a toxicity study and then sometime later after the treatment there might have been toxicities that occurred that we didn't see originally. That is, they developed later. And we didn't have answers to those questions with the studies that we had done. They also said that they did not want us to start clinical trials in patients. They wanted us to start clinical trials in normal volunteers. And the main rationale for that is that you sometimes cannot discern the true safety of a drug if in a patient there are disease-related problems. So the concern of the FDA is a rational one. We felt that there is a patient population that could be tested and tell the difference between a frataxin-related adverse event and a drug-related adverse event. And that would allow us to move more rapidly to test the compound in the patient population it's intended. So those were the two issues that we had with the FDA. It took us a good 18 months to complete the kinds of non-clinical safety and toxicology studies that answered the questions that they had raised about the potential safety concerns of the compound. And at the end of last year, we were able to complete those studies. And the consequence of that was there were no delayed toxicities that we saw, and that the toxicities we did see at the doses above what we expect to use in man were all reversible. And so that was an important findings and things that apply not just to that compound, but to related compounds that we might develop. While we were having this discussion and completing these studies in the United States, we also were talking to the European authorities about doing clinical studies, and their response to our requests to do studies in patients was different, and they understood that perhaps that was an appropriate place for studying this drug. We also provided them with the additional toxicology studies that we were doing, and so we were able to define a way of starting a single-dose clinical study in man in Italy, and we have started such a study, and we're in the midst of that study right now. So we just completed the second cohort. So that study design taking place in Torino is shown on this slide. It includes 4 cohorts or 4 groups of 5 patients each in a single-dose safety study. Again, primarily you're looking at safety, secondary you're looking at pharmacology of the drug, and the third is whether or not you can discern any biological activity of your drug. So you start at a very low dose and you give a single dose. The opportunities for efficacy do not really exist in a single-dose study like this. The opportunity for seeing a biological effect, like a change in the frataxin protein for a brief period of time, is a very important thing to learn, but it's not likely to result in any efficacy for the patient. And so again, this is the balance that one has to take for, I think, all of us being partners in drug development, is that these are important steps to understand the safety and pharmacology of the drug. And it's through finishing these steps that will allow us to get to more complicated studies and to do them correctly to determine whether or not the compound is truly efficacious. So the first 2 cohorts are single dose. They're tested and evaluated for 28 days, and then we can move on to the next cohort. So we finished the first 2 cohorts. We're starting on the 3rd and the 4th. They're a little more complicated. As we get to higher dose, each patient gets 2 treatments a month apart. One is just placebo and one is the drug, and it's crossed over and they don't know which one they got first. So it's an additional way for us to make sure that we can tell the difference between a patient's disease problems and if they have a problem with the drug. Of course, we're going to measure our biomarkers. We're going to measure whether or not we've been able to inhibit the deacetylase. We'll see whether or not that inhibition results in increased acetylation. We'll also measure frataxin RNA and frataxin protein to see if there's been any change. And in work that we've done in collaboration with Dave Lynch's lab, we've been able to discern that we can actually measure frataxin in cheek swabs as well. So as a non-invasive or not very invasive way of getting a frataxin sample, that's an important thing that we'll be measuring as well. So, so far in that study, we have seen no adverse events from our first 2 cohorts that are related to drug treatment, and we have seen good pharmacology of the compound, reproducible across patients, and the exposure is at or above what we would have expected based on our non-clinical studies in rats, dogs, and monkeys. So those are good things, but it's still in the midst of that protocol. So just to, again, make a point that for any company working in this area, and especially for a small company like ours, it's the partnership with others that makes us able to accomplish what we can. And we've been fortunate to have partners with the FARA network, also with NINDS, as you know, have been active in Friedreich's ataxia and still are interested in working in this area, and internationally, both in Australia as well as Europe. Repligen is fortunate to be the only non-European member of the European Friedreich's Ataxia Consortium. There, the European community, their Europe-wide government put $10 million into a clinical infrastructure and basic science program to work on Friedreich's ataxia, and asked us to join with them in that. So it's a global effort to try and identify and develop these treatments. Another thing that we're doing is in small molecule drug development, any good program that looks to be making progress always has to continue to try and find better and better compounds. When we started the clinical development of 2833 3 years ago, we knew that it had things that could be improved on, and we went about through chemistry, a medicinal chemistry program, to find improved compounds that may turn out to be even better than the first generation compound, 2833. And so we synthesized many compounds. We changed different parts of the structure of 2833 to see if we could make it more stable, to see if we could make it enter into the central nervous system easier. That it was more bioavailable, and when you took it orally, more of it got into your bloodstream. And we looked at all of these different features and were able to identify a compound that we thought made improvements in every one of those features, actually. And this compound now gets a nice little name, 3250. So 3250 is being developed as a follow-on compound, and because of identifying this compound that we think is better than 2833 in some of its pharmacology properties, we are interested in being able to develop that compound, and rather than at this state start and stop with that compound in the United States, we're going back to the FDA and talking with them about 3250. So this is 3250, and this just shows again what you saw before. A dose response. Here's the 2833 and here's the 3250. It has very similar potency on the enzymes and in patient peripheral blood lymphocytes as the first compound. However, it's much more stable, and it's 5 times better at getting into the central nervous system than 2833. And this shows the results in one of the mouse models where you can see the level of frataxin RNA is increased by drug treatment. So let me just conclude again by thanking a lot of the people that make up the team that has worked to try and identify and advance these compounds. At Repligen, Vincent Jacques is the project leader for this program, and a lot of the cell biology work is done by Wenyan Miao. And as I've said before, our collaborations with the Gottesfeld Lab, the Pandolfo Lab, and with the FARA organization, especially Dave Lynch's lab, has been important for our progress. Thank you.